So the first thing I need to do today is to perform a technique called Polymerase Chain reaction. This is a common technique that we use in molecular biology to amplify a specific region of a DNA sequence. So I have two samples of DNA that I'm trying to amplify in small fragments so that I can assemble them into a vector and then transform into E.coli, and then the protein encoded by the gene can be expressed within the cell. So the first thing you need to do in the morning is to set up the reaction and then use agarose gel electrophoresis to see if the amplifications worked. And then I will move on to the second part of the day, which I will explain later in the video. So yeah, so let's get some PCR set up. Okay, so I have set up the PCR and made agarose gel to do the DNA electrophoresis. So I've got about 45 minutes to do the next step. So I am going to get some reading done in the meantime. So here goes. So the PCR I did this morning has worked. I've successfully amplified the region of DNA that I needed to amplify, and now I can go ahead and purify the fragments and assemble them into a vector to be then transformed into E. coli. So that's going to be done later this week. So for the second part of the day, I'm going to set up some protein crystallization screens for the protein that I'm working on. And once we get the protein crystals, I'll be able to use X-ray crystallography to determine the structure of the 3D structure of the protein. So that's the goal here. Some information about the protein that I'm working on. It is very oxygen sensitive, so I have to make sure that everything is done anaerobically. And so the first thing I'll do is I'll go get the protein from the storage, which is stored, so the protein is stored under liquid nitrogen. So I'll get the protein pellets from the storage, and I'll put it in a vial, hook it onto a Schilling line, degas the protein, and then they'll be ready to be taken inside a glove box. So that's basically what I'm going to be doing the second half of today. So let's go make some crystals. Finished all my stuff for today. I've set up my crystal plates for my protein, so that's all done, and that's officially the last thing I had to do today in the lab, so that's all done. Now I am going to spend about half an hour to 40 minutes getting some slides together for a talk I'm giving in the next few days. So I'm going to make some slides, so let's get that sorted, and then I can finally go home. So I am all done for the day. I made a few slides for the talk next week. I've had a productive day in the lab, so that was good. So yes, so overall I had a good day. So now what I'm going to do is I'm going to go home. So thank you for watching. I hope you enjoyed a day in the life of a postdoc video, and thank you.